Today's lesson is about oxidative phosphorylation, the most important generator of ATP in the entire process of cellular respiration. Before we talk about the electron transport chain and oxidative phosphorylation, which is the final step of aerobic cellular respiration, let's go back here and review a little bit about what happens before that process takes place. All right, so remember, glycolysis is the first stage of cellular respiration, and that takes one glucose and turns it into two pyruvate molecules, or pyruvic acid. And in the process of doing that, NADH are generated and ATP are made, a small amount of ATP, 2 to be precise. That pyruvic acid is then converted into acetyl-CoA, moving across into the mitochondria. And acetyl-CoA enters the Krebs cycle. The Krebs cycle again generates a small amount of ATP, and more importantly, it generates electron carriers, NADH and FADH2, which are going to be used in oxidative phosphorylation. Again, the most important creator, or producer, I should say, of ATP. Okay, now how does the mitochondria make these ATP? It uses a pretty cool system. It takes the potential energy stored in these high energy electrons of the NADHs and the FADH2s and uses that energy to do work. And the work that it does causes ATP synthase to generate ATP. Okay, so we're going to look at that process now. Now, the key to this is how the electrons move. Okay, and electrons move through the electron transport chain within the cristae of the mitochondria. Okay, so we're inside the mitochondria here. The electron transport chain is right here. You can see it. Okay, and here's ATP synthase. And remember, these are all proteins. Okay, so they're specifically designed to do a job, a specific job within the mitochondria. Okay, now out here, this area right here is the intermembrane space. I'm going to call it IMS, the intermembrane space. And in here, down in this area here, is actually inside the cristae, or the very inside of the mitochondria, called the matrix. Okay, so out here is the intermembrane space, and here is the matrix. All right? Okay. So here we have the electron transport chain. So in order to move electrons through the electron transport chain, these electron carriers, NAD and FADH2, have to donate their electrons that they gathered back in glycolysis and the Krebs cycle. Okay? Now NADH is going to donate its electrons at this point right here, as you can see. Okay? Now, as the electrons move through the electron transport chain, their potential energy stored in high energy electrons is converted into kinetic energy by pumping protons across the membrane. Okay? So remember that these NADHs, because of the electrons that they have, were being chased or followed by H pluses. They were attracted to the electrons. Well, as the electrons move through the electron transport chain, these H pluses are going to move from the matrix to the intermembrane space. Okay? So all these electrons are going to move through, and as that happens, their potential energy is converted into kinetic energy, which pumps the protons from the matrix to the intermembrane space. Okay, now why? Why is all that going to happen? Well, the idea here is that we create a concentration gradient of protons. Okay, and as we know from past experiences, when we have a concentration gradient, again, we have potential energy. Okay, and that potential energy is going to be released, and when it's released, the protons are going to move into the matrix again through ATP synthase, and that movement is going to help generate, so that kinetic energy, potential energy stored in the proton gradient, is going to be released, creating kinetic energy, which is going to move through ATP synthase, which is going to help generate ATP, so that would be chemical energy. Chemical energy. All right, so it's a lot of energy changes as we see here, but they use these reactions and they recycle the energy to allow the mitochondria to do all this work and create the ATP that's required. Okay, so the chemical energy here and ATP. Okay, so the key to take away from this is that these electrons are released. Okay, so NADH is oxidized back into NAD+. And in the process of doing that, protons are pumped, okay, into the inner membrane space, which creates potential energy. That potential energy is going to eventually build up, which is going to allow the ATP synthase to convert that potential energy into kinetic energy and allow it to do work, which is going to cause ADP to turn into ATP, or chemical energy. Okay, so electrons move from high energy levels to low energy levels as they move through the transport chain, which helps create these ATP. Chemiosmosis. Okay, chemiosmosis is quite simply how the ATP is generated. We talked about in the last part how this H plus gradient, proton gradient, is created. So the electrons move through the electron transport chain, which allows this proton gradient to be created. Okay, we have a high concentration of H plus. Okay, that's what that's supposed to be there. Let's make it a little bit better there. High concentration of H plus. All right, that high concentration of H plus is potential energy, and that potential energy is going to move through the ATP synthase when the ATP synthase is ready, or when the concentration is just right, and that movement due to the proton gradient is called chemiosmosis. Okay? Now, that potential energy, again, as we discussed in the previous, is going to be converted into kinetic energy. Okay? In other words, this gated ion channel, ATP synthase, is going to open up and allow the protons through. When that happens, potential energy immediately turns into kinetic energy. These guys, because there's a high concentration here and a lower concentration here, they want to go in, so they do. And in the process, the ATP synthase converts the kinetic energy into chemical energy in the form of ATP. Okay, now, why do they call it phosphorylation? It's a question we should have answered before, probably. But notice what's happening here. We have ADP, which is adenosine diphosphate, being added to a phosphate to make ATP. They call it phosphorylation because a phosphate group is being added to ADP. In fact, anytime a phosphate group is being added to a substance, they call it phosphorylation. Okay? This particular process is called oxidative phosphorylation because oxidation is occurring, and also the oxidizing agent is oxygen. Okay, and we'll get to that in a second. Okay, so again, for chemiosmosis, it's the buildup of a concentration gradient of protons, potential energy that gets turned into kinetic energy. The kinetic energy is converted into chemical energy in the form of ATP by the ATP synthase. Okay? All right. Electron flow and the consumption of oxygen. Let's talk about that now. Okay, so remember, NADH and FADH2 donate their electrons. Okay, they're oxidized. Anytime there's an oxidation reaction, there also has to be a reduction reaction. Okay, and this is the exact opposite reaction as to what happens in photosynthesis. During the light reactions, water is split into protons and electrons and oxygen. Well, in this case, we have the protons that came from the NAD and the FAD, combining with electrons and oxygen that was brought in by the mitochondria to form water. Okay? And we say that oxygen is the final electron acceptor. So notice here, electrons are passed. They're passed from one protein complex in the electron transport chain to another. But eventually they run out of places to go. So oxygen is the final electron acceptor. Now, if oxygen wasn't there, if this wasn't here, then none of this would happen. Because you can't have an oxidation reaction, or two for that matter, without a reduction. Okay, and in this case, oxygen is being reduced to water. It's the final electron acceptor. It's an oxidizing agent. Oxidizing agent. Okay, but none of this would happen if oxygen wasn't there. So these flow of electrons only happens if oxygen is present. Never any other time. That's why without oxygen, the entire process shuts down. And the cell goes into anaerobic respiration. To sum up, let's talk about in total what we get from this, because this is really important. Okay, so for one glucose molecule that goes through glycolysis, as you see here, which happens in the cytoplasm, by the way, you get a total of 2 ATP. Okay? We have from one glucose, we get 2 pyruvates, 1, 2, and those 2 pyruvates create a total of 2 more NADH in the conversion to acetyl-CoA, 8 NADH for 2 turns of the Krebs cycle, and 2 FADH2 for 2 turns of the Krebs cycle, along with 6 carbon dioxide molecules. Again, this is the conversion of pyruvate to acetyl-CoA. Don't forget about that. That's a kind of separate step. You know what? I'm just going to say A, C, C, O, A. Acetyl-CoA. Okay, that's what's happening there. Okay, so through the Krebs cycle, only 1 ATP is generated per turn for a total of 2 ATP. So up until the electron transport chain and oxidative phosphorylation, we've only generated 4 ATP. But during the electron transport chain and oxidative phosphorylation, all those funky reactions we just talked about in the previous sections, we generate 32 ATP for a net harvest of 36 ATP. Okay, so without oxygen, the maximum amount of ATP you're going to get is 2, because the Krebs cycle won't happen and the oxidative phosphorylation won't happen, which means you don't get any of those. So oxygen is key to this process to get the amount of energy we need to actually do the things that allow us to survive for an extended period of time. You can survive on 2 ATP for a little while, but if oxygen isn't brought back into the equation eventually, if aerobic respiration doesn't happen, we're not going to survive very long. We're not going to have enough energy to do all of it, because remember, all of the processes that happen, every single reaction that happens, every time something is actively brought into the cell or put out of the cell, it's going to require a certain amount of ATP. If you're only getting 2 for every glucose molecule, it's not very efficient. 36 ATP, on the other hand, is quite efficient. And that's the purpose, right? That's the whole reason we did this process in the first place, was to generate that cellular energy. Okay, please make sure you go back and review this section again. It's very important that you understand how the electrons move through the electron transport chain. You don't have to name any of the complexes, it's not important, but you have to understand why they're moving through and what happens when they move through. How is ATP generated in oxidative phosphorylation? If you can answer those questions, then you're fine. Make sure you understand where the oxygen fits in as well. That's very important. Please bring your questions to class and make sure you take good notes on this section. I'll check those when you come to class.